Hello there everyone. I thought I'd again continue working my way through the past papers. Today we're going to do the Biology Paper 1, a higher tier paper. Now I've already done the foundation tier of this paper from 2018, and remember that with the foundation and the higher paper there are crossover questions which apply to both. So I'm going to cut out my answering of those questions and make that the start of the video, so don't get too put off by the question numbers. So let's get into it. So moving on to question 6 then. It says cell structures, draw one line from each cell structure to the type of cell where the structure is found. Okay, so nucleus is eukaryotic cells. Eukaryotic cells have nuclei, and what we call membrane-bound organelles. Now, that's a fancy word, but nucleus is a membrane-bound organelle, but that basically means mitochondria. That means vacuoles, and that means chloroplast. So permanent vacuole has got to be a eukaryotic cell, but a permanent vacuole is actually only in plant cells, and plant cells are eukaryotic cells. Plasmids, I like to think about them as DLC for games. Okay, so obviously you might be familiar with this, but you buy a game. So here's my game, and then you play the game, and then they might release a little extra add-on pack, DLC, downloadable content, where you pay a little bit extra money and you get a little bit more game. So in bacteria, you have this chromosomal DNA. That's the main kind of DNA of the bacteria, and then you can have these little extra bits of DNA which can add on little extra genes which maybe could carry antibiotic resistance and other things which are useful to the bacteria. So like a game and its DLC. So plasmid is only in prokaryotic cells, and bacteria are your big example of these. So they don't have a nucleus. Notice how I drew that as a loop of DNA, not a nucleus. Okay, that is the DNA floating about on its own. So that's bacteria and stuff like that. Moving on. Figure 11 shows a plant cell. What are the names of structures A, B and C? So A is pointing to the vacuole. So I know that it can't be this row because it hasn't got that there. And now I've got to decide here. Now C is pointing to the right outside. So this is a plant cell. So the right outside of a plant cell is the cell wall, not the cell membrane. But just to clarify, we'll just make sure of this. B is pointing to the smallest structure I can see here. And the smallest structure in a plant cell, or most eukaryotic cells, is a ribosome. They're very, very tiny. Can't see them with a light microscope, need an electron microscope. So that means the bottom row is the one that we're after. The student observes slides of onion cells using a microscope. Figure 12 shows 2 of the slides the student observed. Here it's a little bit fuzzy, but you can see they're much bigger than they here in slide B. The cells on the slides are not clear to see. Describe how the student should adjust the microscope to see the cells on slide A more clearly. So hopefully this is really asking you about microscope technique, and asking you how do you focus a microscope. And there is hopefully you'll remember that there's a kind of a focusing knob that you can turn, and that will adjust the height and therefore focus it. Because this is so zoomed in, I don't think it needs that much. So I'm going to say you could turn the fine focusing knob, because there's a coarse one where it does it really quickly, and there's a fine one where it's a little bit more slowly. Okay, until the cells are in focus. Because, I don't want to go past that. The cells are in focus. Okay, 6.4. Describe how the student should adjust the microscope to see the cells on slide B more clearly. So this is in focus, but you can see it's not as zoomed in as this. So if I was wanting to see individual cells more clearly, I would switch to a higher power. Now, if I'm looking at a microscope, you have two lenses. You have here, you have the eyepiece lens, is what you look through, and then you will tend to have this kind of thing here where I've got kind of three options, and you're only really looking through one. Okay, at my slide like this. This is the worst drawing of a microscope ever. You can tell I didn't do art GCSE. So this one might be 4 times, this one might be 10 times, this one might be 40 times, or something like this. So what I can do is I can rotate this and change to a higher power. So I would change the objective. So this is these lenses here are called the objective lens. To a higher power. Okay, continue on. Lovely. 3 pages left. Time flies when you're having fun, you know, because this is what I like to do in my free time. He says, the student made the necessary adjustments to get a clear image. Figure 13 shows the student's drawing of one of the cells. The real length of the cell was 280 micrometers. Calculate the magnification of the drawing. So, this is now we're in the questions which appear both on the foundation paper and the higher paper. So this is where they might now start to ask you things where you have to remember them a little bit more. So there's only really one major equation that we are faced with in biology, and that's the magnification equation. So that states that magnification equals image size divided by real size. Okay, so I know the real length is 280 micrometers. Okay, that micro is going to be important because this only works if they are in the same units. And I don't even need to get my measurement out, but actually, you know what? I just fancy checking the exam, see if they're right. I tell you what, there you go, 112 millimeters. Look at that, beautiful. So I'm going to put it into this equation, but the first thing I'm going to have to do is change either the micrometers into millimeters or the millimeters into micrometers. I'm going to change the millimeters into micrometers. Now, unit conversion is one of the hardest things that you have to deal with, really, and it relies on us knowing the unit system. Okay, in the metric system, we realised that it's much easier to work in tens, okay, or thousands, where it's a nice round number and the units scale really nicely. Back in the old days when you had imperial, and obviously some of you'll still be familiar slightly with imperial, there are, for instance, 3 feet in a yard. There are 1,760 yards in a mile. There are, but you could also have 8 furlongs in a mile. It gets really weird, okay, and the units get really bizarre. So what they said is we'll make everything on nice powers of 10 or /1000, okay. So what we're going to do is we're going to take the unit meters, because that's the unit for length. And if I'm dealing with smaller lengths, I'm going to add something at the front, okay, which tells me I'm dealing with smaller lengths. So I'm going to add another m, and it doesn't actually matter what the units are. If I add a little m at the start, so if I had litres, for instance, and I had ml, that's millilitres. If I had grams, g, it'd be milligrams. And what that little m at the start tells me is that it is 1,000 times smaller. Okay. If I want to go 1,000 times smaller still, now this unit is a bit weird. That is the Greek letter mu, and that's the Greek letter m, so it's like an even smaller number. And that is 1,000 times smaller still. Okay. So, if I've got 280 micrometers, right, so I've got 280 of these, and I want to convert it up to millimeters, that means that because each one of these is 1,000 times smaller, that means that I'm going to have to divide by 1,000. So 280 micrometers is going to be 280 divided by 1,000, which is 0.280, I suppose, millimeters. So now I've converted it, I can put it into the equation. So magnification equals our image size, so that's the size of this image, what I've just converted. I haven't just converted it, it's already given to me in 112 millimeters, divided by my real size, which is what I have converted over, and is 0.280. And I can put that into the calculator, use a calculator. Remember, there's no non-calculator science exams. I'm going to use Siri again. Hey Siri, what's 112 divided by 0.280?
112 divided by 0.28 is 400.
Thank you. And that means that my image has been magnified 400 times. Okay, good rule of thumb is your magnification is always going to be above 1. If it's smaller than 1, the question's been very mean, but if the magnification is smaller than 1, the chances are you've probably got the numbers the wrong way round, and you've put the real size at the top and the image size at the bottom. Okay, last question. Coronary heart disease is a non-communicable disease. Coronary heart disease is caused when fatty material builds up in coronary arteries. Explain what a non-communicable disease is. So if you look at the word communicable, that to me shouts communicate, right? And when you communicate, that means that you are spreading information. You know, if I'm talking or communicating with someone, I'm spreading the information of what I'm saying to that other person. So if I've got a non-communicable disease, that means that I can't communicate it, I can't spread it. Okay, so a non-communicable disease is one which cannot be passed on. And why can't it be passed on? Well, that's because it's not caused by a pathogen. Remember earlier we discussed the pathogens and we said about bacteria, virus, fungi and protists. They are little things which can be passed on. Okay, so if it can't be passed on, it means it's probably not going to be caused by a pathogen. So non-communicable disease is one which cannot be passed on. This is because it is not caused by a pathogen. Okay, figure 14 shows a coronary artery of someone with coronary heart disease. And you see the artery wall here and the fatty material built up. Okay, so explain how that can cause a heart attack. So if we're looking at the diagram, you can see here that that space is much smaller because of that fatty buildup. So if I'm restricting that flow of blood, that means that I'm going to get less oxygen and other nutrients to the heart, and that means that the heart muscle, which has always got to work, okay, is going to get less oxygen to it, so it means it cannot respire, do that respiration to get that energy to contract. Can't do that as much as it needs to, and that means that heart muscle will start to die, and that is what we cause a heart attack. So coronary heart disease reduces blood flow through the artery. Okay, this means that less oxygen can get to the heart muscle. This means not enough oxygen for respiration, and the heart muscle will begin to die. And that's what a heart attack is, where part of your heart has started to die. The last question is a nice 6-mark question to finish. What a treat. It says, explain how lifestyle and medical risk factors increase the chance of developing coronary heart disease. Okay. So this is a long answer question, so we're going to use Rustman to answer it. Okay. Remember, these are purely advisory. It's not like a step-by-step process. It is in some regards, but it's a little bit more vague than a prescribed plan. So I'll just read the question. So I'm going to see here, it says, explain how lifestyle and medical risk factors increase the chance of developing coronary heart disease. Underline the command word. So I'm going to look at explain. That means I need to say why those things are important. Subject words in here. We've got lifestyle factors, we've got medical factors, and coronary heart disease. Think of any other keywords. I'm thinking straight away things like obesity. I'm thinking things like diabetes, exercise, alcohol, things like genetics. These are some other keywords that maybe I'm going to want to mention. Make sure you organise your answer. So I'm going to take it in 2 paragraphs, and I'm going to look at the lifestyle factors and the medical factors, answering all the question. Again, this is really important in a question like this, because if you choose only lifestyle factors and only medical risk factors, then you're excluding one half of the answer, and that means you're not going to be able to get all the marks available. And then once we're done, we're going to go back and check. So lifestyle factors, okay, include things like smoking, because that's a choice, isn't it? Okay, you are choosing to smoke. Things like obesity. It's a little bit more vague, but obviously if you eat too much, then that's not going to help. Things like lack of exercise. Things like a high fat and salt diet. And one more might be air pollution. So for instance, you get more coronary heart disease in urban areas. So medical factors, so I'm going to start a whole new paragraph, so I'm organising my answer, include things like high blood pressure. You might not be able to help that. That's a medical thing, isn't it? You don't go, I'm going to have high blood pressure today. High cholesterol. Things like diabetes, as we said, and things like and genetics. Okay, you don't choose the genetics that you're born with. Notice how, if I stopped here and I read back, I'd notice that I haven't actually answered the question, because I've just stated those factors, I haven't explained. So I'm going to add one more paragraph just to explain how they actually increase the chance. So obesity and high cholesterol. Notice I'm picking one from each there. Can cause fatty deposits. Those things that we saw on the previous question, building up in the arteries, and coronary heart disease. Things like smoking and high blood pressure. So smoking, and actually we're going to go with high salt diet can cause high blood pressure. And if you think about it, if I'm really pumping the blood through that small cavity, then that means that some of this fatty material might break off and actually cause a heart attack, which increases stress on fatty deposits, leading to heart attacks. And one more is I'm talking about the genetics. Okay, genetics can affect how easily you develop coronary heart disease. Okay, that's not the best answer, but I think I've hit all of the points. So just reading back, lifestyle factors include smoking, obesity, lack of exercise, high fat and salt diet, and air pollution. Medical factors include high blood pressure, high cholesterol, diabetes, and genetics. Obesity and high cholesterol can cause fatty deposits and coronary heart disease. Smoking and high salt diet can cause high blood pressure, which increases stress on fatty deposits, leading to heart attacks. Genetics can affect how easily you develop coronary heart disease. Again, it's not the best answer. If I had a mean exam, I probably might not even get full marks, because it's weird. These 6-mark questions are very weird to mark. But notice by following that, I've increased my chances of getting those high marks by making sure that I organise my answer, making sure I'm including keywords, and making sure I'm answering all the questions. So that is the 2018 foundation paper. I hope you found that useful and not too boring. Okay, so this is our first question, which is only on the higher paper. This question is about photosynthesis. What is the correct balanced equation for photosynthesis? Now, firstly, we need to make sure that we've got the right thing on both sides. The word equation, which some of you probably find easier to remember, is carbon dioxide plus water goes to glucose, which is a form of sugar, plus oxygen. Okay, so we need to make sure that we've got those on the right sides. The formulae of these are carbon dioxide is CO2, water is H2O, glucose is C6H12O6, and oxygen is O2. So if I have a look, the only ones which have got that on the right side is this one here, the third one. And notice how I didn't even worry about the balancing. It's the only one where the actual symbols match up. If you are asked to balance it, just remember that this is an absolute devil to balance, and you can just put 6, 6, 6, and it will balance. So that's the correct answer. What type of reaction is photosynthesis? Aerobic, endothermic, exothermic, or oxidation? These aerobic is all to do with respiration, really, and if it requires oxygen. This doesn't. It's not got oxygen on the left. It's not a reactant. Again, oxidation is the gain of oxygen or loss of electrons, and it's not doing that. Endothermic or exothermic. Now, these are all talking about energy in or energy out. Endothermic, think of the word enter, and exothermic, think of the word exit. So am I getting energy out of this, or am I putting energy in? Well, photosynthesis requires energy in the form of light. Okay, so if it's taking in energy, then that will be an entering of energy, and it will be endothermic. Looking at the next one, it says the student investigated the effect of light intensity on the rate of photosynthesis. Shows the apparatus used. Sodium hydrogen carbonate solution releases carbon dioxide gas for the pondweed. You can see that they've placed it at 5 centimetre intervals. They've then measured the rate of photosynthesis by counting the number of bubbles produced. Repeat with the pondweed at 10 centimetres and 20 centimetres from the light source. It's not an accurate way. Suggests 2 ways the method could be improved to measure it more accurately. Okay, the first one is if you do it over a longer period of time, then you're going to get a better average. So the first thing that you could do is measure it over a longer period of time. Okay, and another thing that we could do is instead of, if you think about it, bubbles are of different sizes. You can even see it here in the diagram. So I could measure the volume of gas given off. So both of those ways would be a little bit better than what I actually have. Okay, the LED light source does not get hot. Explain why it is important that the pond weed remains at a constant temperature. This is all to do with control variables and independent variables. In this experiment, they're looking at the effect of light intensity only. That means the only thing that I want to change is the light intensity. So my independent is light intensity. Okay. If the LED gets hot, okay, then that means that the temperature is going to change. It means it's not going to be a constant temperature. Now, it says here why. Explain why. Okay. So, if I change the temperature, I'm going to change the rate of photosynthesis. That's my first reason. But then thinking of the why is, why does it affect the temperature? Okay, and that's all to do with the enzymes which perform photosynthesis. So, if the pondweed is at so if the pondweed is at a higher temperature or different temperature, then it will have a different rate of photosynthesis. Because remember, temperature is a factor which affects it. Now, why does it affect it? For that why part of the question, this is because photosynthesis is controlled by enzymes. And that covers our why part of the question and gets that second mark. Light intensity can be calculated using the inverse square law, where L is light intensity and D is the distance from the pondweed, distance of the pondweed from the light source. The student placed the pondweed at 5, 10 and 20 centimetres from the light source. Explain how light intensity changes as the distance of the pondweed from the light source is doubled. Okay, so it says you must include calculations in your answer. So the easiest way is just to pick two distances here and put the numbers in, and pick two of them where one of them is doubled of the other. So for instance, let's look at 5 centimetres. So at 5 centimetres, the light intensity will be proportional. So it's not going to be exactly this, but it'll be proportional to 1 over 5 squared. Now, admittedly, I probably could have put that into metre squared, but again, this isn't 100% accurate. So by having that proportional, it means that's fine. As long as we keep our units consistent between the calculations, we don't need to do any conversions here. So that will be that will tell us that light intensity is proportional to 1 over 25. If I do 10 centimetres, remember it's double, then that will be proportional to 1 over 100. Now you can see here the length has, the distance has doubled. Distance has doubled, D is doubled, but the light intensity has got 4 times smaller. Okay, smolter. It's got 4 times smaller. So, as the distance doubles the light intensity gets 4 times smaller. I really can't spell smaller today, can I? Okay, or a quarter of it, I suppose. So, 3.6. The student's results are shown in Table 1. Predict how many bubbles of gas would be produced in 1 minute if the pondweed was placed 40 centimetres from the light source. Okay, so this is going to rely on what we've just done in 3.5, where we've come to the conclusion that if I double the distance, the light intensity is 4 times smaller. So if there's 4 times less light, then that'll be 4 times less photosynthesis, is our logic. Okay, so if 20 has produced 8, then 40 is double 20. So we would predict 2 bubbles, which is 4 times smaller. Okay, this is because there is very little light for photosynthesis. So we'd expect it to be smaller, and using what we've done in the previous question, we've expected it to be 4 times smaller. Describe how the student could change the method to investigate the effect of carbon dioxide concentration on the rate of photosynthesis. You should include how to change the independent variable and 2 control variables. Okay, so if we think about what affects photosynthesis, okay, we've talked about light intensity, we've talked about carbon dioxide, we'll talk about temperature. Okay, so here I'm now wanting to look at carbon dioxide concentration. So this is going to be the one that I'm going to change. This is my independent variable, and these 2 are going to be my control variables. So 2 things which immediately we're going to want to control for are light intensity and temperature. Now, what was it in this experiment where which gave me the carbon dioxide concentration? Well, it's the sodium hydrogen carbonate solution. It says here, sodium hydrogen carbonate solution releases carbon dioxide gas. So, if I'm going to change a carbon dioxide, I'm going to do the same experiment, but instead of 0.2, I'm going to vary the sodium hydrogen carbonate solution. How are we going to control for light intensity and temperature? Well, temperature, I'll just make sure that the solutions are all the same temperature. For the light intensity, I pick a distance and I stick with it. Okay, so I might say 5 centimetres. Okay, so to vary the carbon dioxide concentration, we would change the concentration of sodium hydrogen carbonate solution. So that's the first part of the question, and again, just to make sure I really signal to that examiner that I know what I'm doing, I'm going to just make sure that my next part, the control variables, are on the next line. So how am I going to control them? So to control light intensity, I would use the same distance. I would also control temperature by ensuring all solutions at the same temperature. Okay, And that was a really prime example, that question, of you'd worked out lots of parts for later questions earlier on. Like here in 3.6, we've done stuff of it in 3.5. Here, we needed to turn back to the experimental method. Okay. Question 4. A student investigated the effect of different concentrations of sugar solution on pieces of carrot. This is the method they used. They weighed 5 pieces of carrot. They put each piece into a different tube. Into each tube they added 20 centimetres of water, or one of the sugar solutions. Oh, I can't say sugar solutions. As shown in figure 6. They left the apparatus for 2 hours. They then removed the carrot and dried each piece on a paper towel. That's important because I'm removing that water from the outside. And then reweighed each piece, and I calculated the percentage change in mass of each piece. Okay, so the first question says, why did they pick a percentage change? Well, that's because I've got 5 pieces of carrot. Now, it's going to be very problematic getting the same mass of carrot to start off with. So the easier way is to look at the percentage change, because then what I've done is I've taken into account the different starting masses of the carrot. Okay, this is because the carrots had different starting masses. So by doing the percentage change, I've controlled for that. Okay, question 4.2 is a graph question, and whenever you see a graph question in the exam, you've got to be happy, because you get 4 marks for these. The key thing here, whenever I'm looking at line of best fit, okay, is remember that it's not like maths. Okay, in maths they say it has to be a straight line, so it could be straight line, and it could also be a curve. Okay, these are your 2 options. It could be either or. Don't force a straight line on a curve, and don't force a curve on a straight line. It says choose a suitable scale and label for the y-axis. That's always the hardest part of plotting a graph, and I would say is what separates out people. The osmosis graph, I think, is actually one of the hardest graphs to do at GCSE, because it goes negative. Okay, because you can have a look here. I've got to plot up to plus 24, and I've got to plot all the way down to minus 15. So I need a scale which represents that. Well, the first thing I'm doing when I'm crafting a scale is I'm looking at the number of big squares, I call them. Okay, big squares on the graph paper. I've got 1, 2, 3 below, and I've got 1, 2, 3, 4, 5 above. So if I used 1 to 1 big square, I clearly am not going to get high enough, because I'd only be able to get up to plus 5 and minus 3. Okay, so 1 doesn't work. Let's try 2. That would be 2, 4, 6, 8, 10. Minus 2, minus 4, minus 6. Well, that's not enough. Don't try 3, because thirds is annoying. 4, would that work? 4, 8, 12, 16, 20. Not enough. Okay. Clearly, and probably some of you are already thinking this, it's quite obvious, it's going to be 5s. Okay, but that methodical approach is what's going to be easiest for slightly harder scale questions. So, 5s does fit. So if I have a 5 here, 10, 15, 20 and 25. Now it's a bit rubbish of me, because I should really be using a ruler and pencil. I will use a ruler, but I'm not using pencil. Sure, it shows up a little bit easier on the camera. That's my scale. Very often people miss that label. So what is it? It's the percentage change in mass. Now, label the axes doesn't need to be difficult. It's just the column headings. Okay, and you can see that this one over here is your x-axis label. Okay, now plotting the results. So at 0, okay, so when it was in pure water, it had a percentage change of 24, so I'm going to go up to 24. Using my scale, it means 2 little squares is 1, so 24 is going to be 2 little squares from 25, which will make there. 0.2, so it's got a little bit of sugar in now, is at plus 12. Okay. 0.4 is at plus 1. 0.6 is at minus 8. Okay, and 0.8 is minus 15. Now, this question was actually controversial when it was in the exam, because lots of students complained because they said that we haven't done any practicals with carrots. They have, okay, they've done it with potatoes, and there's a bit on the specification which says that you can use any root vegetable. What I've drawn there is a line of best fit. Now, it's not perfect, but it's pretty fair. When you're in a line of best fit, what you're looking for is you're looking for the rough shape of the points, and you can see here that they're all roughly on a straight line. You're then looking to get the same amounts kind of left and right of the line. So you can see here I've got kind of one above by a little bit of a distance, one above by a little bit of distance. These two are quite close below, and this one's a little bit further away, so they kind of balance each other out. Trust your instincts a bit, okay? The line of best fit is exactly what it says on the tin. It is a line that best fits the points. You're looking at the trend, so I wouldn't worry too much if your point is a little bit off. Okay, estimate the concentration of sugar solution inside the carrot cells. Use your completed graph. Now, this is something that you can work out by doing this experiment, is you can work out the concentration inside. Now, if you remember osmosis, it relies on concentrations of water inside and outside. Now, if it's gaining mass, that means water's moving into the carrot. So this section above the graph here is where water's moving into the carrot. These kind of concentrations here, okay, where mass is moving out of the carrot, okay, here, that means that the concentration of the concentration of the sugar solution is such that water is moving out of the carrot. So the concentration of the carrot will be where it's neither moving in nor it's moving out, where the outside solution perfectly balances the inside. So it's in kind of an equilibrium. This is also referred to as an isotonic solution. So we're looking on the graph for the point where water neither moves in nor out. And you can see here, according to my line of best fit, that occurs where it crosses, where I've got 0% change in mass, and it's between 0.4 and 0.6. And this line here would be 0.5. It's not quite there. Okay, it's 3 little squares and a little bit. So that would be probably about 0.47 to me. Okay. It then says, explain why the mass of the carrot in the 0.6 mole sugar solution changed. Okay, so this is explaining the 0.6. So I'm going to look here, and you can see here that the 0.6 has lost mass. Okay, so the carrot has lost mass. That's my first point. So I've now got to explain why that has happened. This is all due to osmosis, as I've said. So if I think about this here, okay, here's my piece of carrot. Now inside here, I've got things like sugars, salts, bits of cell. Okay, and I've got water. Outside, because it's 0.6, that means that actually it is more saturated, it's got more sugar and in it. Okay, so if we remember what osmosis is the movement of water from an area of high concentration to low concentration of water. So it's high water, or you can think of that as low sugar. to low water concentration or high sugar. So looking at this example then, I've got outside, so this is the carrot, inside the carrot I have got high water, low sugar, and outside the carrot I have got high sugar, low water. So that means the water will move out of the cell, which is what we expect because the carrot has lost mass. So how can I put that into nice words that I can talk about? Well, basically this question is asking me to explain osmosis. Okay, so why has the carrot lost mass? Okay, this is because of osmosis. Well, this is due to osmosis. Okay, what is osmosis? Okay, this is the movement of water from a higher concentration of water, which we've said is inside the carrot cells, okay, to outside not to outside, to a lower concentration of water, which we've said is outside the cells or outside the carrot. Okay. Now there's one other thing which is necessary for osmosis, and that's the idea of a partially permeable membrane. Okay, that's a membrane which only lets some things through. In particular, it will let water through, but not the sugar. Okay. It also requires a partially permeable membrane, and this is what the carrots have, which carrot cells have. My handwriting went a bit rubbish there, but there you go. Okay, so they repeated the investigation with boiled pieces of carrot. Suggest why the pieces of carrot did not now change in mass. Well, it's all to do with this partially permeable membrane now. Partially permeable membrane. Because if I boiled the carrot, I'm probably going to be damage that cell. Okay, so I'll probably burst them. You know this because if you ever boil vegetables, you get colour in the water, right? You know, if you boil peas, the water turns a little bit green. So that means that the membrane's broken apart and some contents of those cells are spilled out into the water. So, it suggests why the pieces of carrot do not change in mass. This is because the high temperature damaged the partially permeable cell membrane. Okay, that's question 4. On to question 5. The measles is a serious disease. That's a nice glib sentence to start off with. A person can die from the measles. Table 3 shows the number of medically confirmed cases of measles in England and Wales between 2012 and 2015. Yeah, another example of Scotland being left out. Well, they have their own NHS, that'd be why. So did Wales, though, I think. Anyway, the year 2012, they've got that number, and it's going down. So just one reason why the actual number of cases of measles in England and Wales might be higher than is shown in Table 3. Okay, what a perfect environment to talk about this now, right? When we're getting daily confirmed coronavirus cases. Okay, this is medically confirmed cases, right? With coronavirus, you are medically confirmed if you take a test and you're positive coronavirus. And you will go and get a test if you suspect that you've caught it or you're showing symptoms. So not everyone's going to be going to the doctor for coronavirus and getting coronavirus tests. Same thing with measles. Not everyone is going to be getting ill enough and go to the doctor. Okay, and that means that not everyone will be getting a test. Those samples might not often be sent for testing for measles anyway. Okay, so the actual number will always be higher than the number of confirmed cases. Okay, not everyone might go to the doctor and be confirmed measles cases. I will say this: the exams in the future are going to feature coronavirus a lot because examiners like to bring up relevant examples. They like to feel relevant. Question 5.2. Calculate the percentage decrease in the number of medically confirmed cases of measles between 2012 and 2015. So, percentage decrease. Okay, so we're looking for the percentage change in something. Okay, you should hopefully remember this from maths, is the change over the original. Okay, times by 100. So, if I look at the number of cases, I can see that in 2012 there was 2,030, so my original is 2,030. My change, well, I've still got 91, so I've decreased by 91, and 2,030 minus 91, okay, is 1,939. Okay, and if I work that out, okay, it comes to around 4.5%. Okay, you can have it's not going to be 4.5%, more. Okay, and this is why you always check your answers and you always use a calculator. Hey Siri, what's 1,939 divided by
1939 is an integer. 1939 is also...
This is what happens when you work with machines.
Sorry?
What is 1,939 divided by 2,030 times by 100?
Here's what I found.
You know what? Siri's sacked. Siri's fired. Okay, it'd be 95.5%. It says a lot that I can kind of do that without having to worry about her. Use a calculator. Don't use Siri, not allowed to phone an exam. Use a calculator. Okay, so that's the percentage decrease. And yeah, that's it. So I can write that there. And that gives me my 4 marks. Okay, moving on. One reason for the decrease in the number of cases of measles is that more children were vaccinated against the disease. Vaccinating a large portion of the population reduces the spread of the measles virus. Explain why. Again, this is the perfect time to be talking about viral diseases. Okay, if more people are immune to an infection, then there are less people susceptible to the infection to spread it. There are less people who can catch the disease and then pass it on to someone else. So if I vaccinate more, then that means that those people who aren't vaccinated are less likely to come up against someone who has caught the disease. So those people, even if they haven't been vaccinated, okay, have what we call herd immunity against the disease. Now that phrase has got a lot of loaded connotations at the moment because there is implications that the UK government followed that early on, saying that if enough people caught coronavirus, then other people can't get ill from coronavirus because they've already caught it. Okay, whether that's right or not, if they backtracked, whatever. Okay, I'm not going to get into the big discussion. But that's because we don't have a vaccine for it. We can create herd immunity for diseases we have a vaccine for, because we can make people immune without them having to catch the disease. Okay, so by immunising people, that means that most people will not become ill from the disease, and that means that there's less chance of unvaccinated people coming into contact and being exposed to the pathogen. Okay, so vaccinated people are immune or less likely to get ill from the disease. Okay, this means unvaccinated people are less likely to be exposed to ill people and catch the disease. Okay, figure 8 shows the concentration of measles antibodies in the blood of a boy. Concentration of measles antibodies in blood in arbitrary units, and you can see here the vaccine's injected. Okay, you get like this, and then they got exposed to the measles virus, and the concentration went skyrocketing up and went down. Explain the differences between antibody production after the vaccine injection and after exposure to the measles virus. You should include data from figure 8. Okay, so again, I'm going to bear in mind a little bit of Russman that I've talked about before. Okay, where I say that these are some guiding principles to keep in mind. R is read the question, so that's exactly what I've done to start off with. Okay, I've had a look and read through. You underline the command word, that's explain. So I've got to suggest why that is the case. Subject words: antibody, big one. Exposure, measles virus, vaccine. Think of any other keywords. So I think those are the main ones, really. Okay, the other one you can think about is what actually produces antibodies. Well, that's white blood cells. I'd say that I'm going to have to talk about white blood cells at some point. M, make sure you organise your answer. Well, I think I'm going to have to take this in two halves. I think I'm going to look at the differences between them and then explain. So I'm going to kind of just do a bit of a describe and explain. A, answer all the question. We've got to include some data from this graph, okay, as well as making sure that I explain the differences, not just stating them. Okay, and afterwards we'll make sure we go back and check. So, if I have a look here, the number of antibodies after the measles virus exposure is much more than the vaccine injection. Okay, so after the measles exposure, the number of antibodies produced, or concentration, I suppose I should say, and I'm going to quote it from the graph, that is 7.2, was much higher than after the vaccine. And that is 0.8. Okay, so I've used some data from the graph, that's good. What other differences can we see? Well, if you have a look here, okay, I've got this flat time, so I got exposed to the virus, and then there's a flat peak, flat bit before the line goes up. So I've got some lag time before the vaccine produces antibodies. So after I've been vaccinated, I don't get that lag time. Can you see? I get exposed to the measles virus and boom, immediately up. Okay, so also the antibodies were created much sooner than after the vaccine. Okay, where there was a lag. Okay, so now I've done the kind of describe. Now I need to make sure that I'm answering that explain. What is it that my question is? What scientifically is going on here? I need to give a reason. So that's what my second paragraph is going to do, and I'm going to actually leave a line again, making sure that I structure my answer, make it really clear to the examiner in the very small time that he or she has to answer your question. Okay, that they can look at it and give you the marks really easily. So why is that the case? Well, here after the vaccine, okay, our body is learning about the virus and learning to develop, okay, the antibodies for it. Okay, and here we don't have that lag. So that's going to be because I've learned how to make those antibodies. So we have in the immune system specific white blood cells. We have memory cells which remember how to make those antibodies. Okay, so antibodies are made by white blood cells. After the vaccine, the white blood cells have they've kind of learned, but I don't want to think about them as like a conscious process. I'm going to go remember how to make the specific, because remember antibodies are specific to each pathogen. Measles antibody using memory cells. That's not like memory cells in your brain, okay? They are specific, the cells which made the antibodies in the first place, okay? They kind of continue to exist. So this means the response can be immediate, large, and specific. And that explains what's going on. So notice I've followed the principles of Russman there. I read the question, I then underlined the command word. I had a look at what keywords are going to come in, looked at the idea of white blood cells and memory cells, structured my answer nicely with making sure that I organised my answer, and I answered all of the question because I've included some data, and I've gone back and read through my answer. Okay, question 6. Last question. This question is about stem cells. Give one place in a plant where stem cells are found. Okay. Now, if you just say stem, that's not enough. Okay, you need to say memory stem. Not memory stem. I'm thinking of the last question. Meristem. That's one place where stem cells are found. Also, at the kind of the tips of any shoots or roots. Okay, so you can kind of take cuttings and put them in, and they will turn into anything. Or if you think about it, those are the parts of the plant which are growing, and that's what stem cells do. They grow and repair. So I'm going to say meristem. What is one economic use of plant stem cells? Okay, so stem cells divide into identical cells. So if you think about it, if I've got a plant stem cell, I can allow it to grow and make identical plant cells. So if I'm looking at the advantages of plant stem cells, what I'm really looking at is there any advantages to having the same plants? Well, to create genetically modified crops, that's a different process, so it's not going to be that one. To create a new species of plant, while I'm keeping the genetic information the same. To increase variation, again, I'm keeping that genetic information the same. Produce large numbers of identical plants, yes I can, because I can grow those stem cells, and then I can take cuttings and I can make a large number of identical plants. And that's what they do, okay? All when you go to the shop and you buy apples, okay, you buy a specific type of apple, and that is a clone from that original apple type. So all Granny Smith apples come from the same Granny Smith tree. Embryonic stem cells divide by mitosis. Figure 9 represents the cell cycle of a human embryonic stem cell. The mass of DNA in the cell at the start of the cycle is 6 picograms. A picogram is 10 to the minus 3 nanograms. Convert 6 picograms to grams. Give your answer in standard form. Well, this is nice. The reason it gives you this part is because nanograms is part of the specification, i.e. you need to know how small it is. But a picogram is not, so they're saying that it's 1,000 times smaller. Okay, so if I think back to how the system works, okay, we've talked about this idea that the metric system introduced the idea of scale. So I have one unit, and then I can add a prefix which changes how big it is. The first smaller one is M for milligram. Then I have a microgram. Then I have a nanogram. And then finally, all the way down here, I have the picogram, which makes sense, right? Because I'm looking at the mass of DNA in one cell, so it's going to be tiny. Okay. Now, what actually changes each time? Well, each suffix is 1,000 times smaller. Okay, so 1 milligram is 10 to the minus 3 grams, or 1 times 10 to the minus 3 grams. If I'm getting 1,000 times smaller still, then I'm going to add another minus 3 to the standard form. Okay, because it's getting smaller, and if you think about standard form, okay, the minus number means I'm getting numbers below the decimal point. Okay, that is that number. Okay, so if I take 1 and I divide by 1,000 to get 1,000 times smaller, I have 1,000th, and that's what this is, and that's what that number is. So if I'm getting 1,000 times smaller again, I'm getting 1,000th of 1,000th, or a millionth, which would be 0.0000001, and that would be 1 times 10 to the minus 6. Okay, 1, 2, 3, 4, 5, 6 hops. Here, 1,000 times smaller again. So it's 1,000th of a millionth or a billionth. And finally to picogram, and it's told us in here that it's 10 to the minus 3, but I'm following this anyway. So that's 1 times 10 to the 12th. Now I haven't got 1 picogram, I've got 6 picograms. So that means I've got that times 6. So it's going to be 6 times 10 to the minus 12. And that is my answer. Mass of DNA is 6 times 10 to the minus 12 grams. That's quite tough. 6.4, the time taken for this cell cycle to complete is 15 hours. Calculate how many hours the cell spends in mitosis. Give your answer to 3 significant figures. Again, this is more of a maths question. Okay. if you have a look here, they've specified that this is 28 degrees of this circle is spent in mitosis. So we need to work out what proportion of this circle is spent in mitosis. Well, remember, a whole circle is 360 degrees. So that means that 28 of those 368 degrees is spent in mitosis. Now, the whole cycle takes 15 hours. So if I times that by 15, I will get my answer. Okay. Now, if I work that out on a calculator, okay, again, series fired, so you have to use a calculator, use it every time, you'll actually end up with 1.1666666 recurring. Now, my big bugbear is when people leave it as recurring. Okay, round it. And it even says here to give it to 3 significant figures. So that'll be 1.17, because the next digit here will round up to that, hours. Now, I don't need to convert it into minutes, don't need to do anything like that, it just says hours, 1.17 hours. Done. Describe what happens at each of the 3 stages of the cell cycle. Okay, whenever I teach mitosis and stuff like that, I always like to think about I think it's really logical. And if I asked you to make take this exam paper and turn it into 2 identical exam papers, the first thing that you would do is you would go to the photocopier and you'd copy every page. Okay, and that's what the cell does. So here's my cell, and here's my nucleus, and all my other organelles. Okay, the first thing it does is it duplicates everything. So you replicate the DNA. So I've got 4 chromosomes in here, so I've now got 8 because I've replicated them, and I've doubled everything else. Right? From now on, I'm not going to draw that everything else. Okay, so it's clearly growing during that time, and I'm replicating all my DNA. Now, the next thing that I'm going to do, if I'm going to copy my exam paper, is I've copied all of the pages. So my motorcycle goes past. I'm going to copy all my pages, and then, because I've probably done that in a bit of a hurry, a bit of a fluster, all of my pages will come out of the photocopy in all different orders. So first thing I'm going to do to make sure I kind of get it right is I'm going to line up my copies. So I'm going to say, here's page 1 of this, here's page 1, here's page 2, here's page 2, here's page 3, here's page 3, here's page 4, here's page 4. So I know that I've got the right pages in each side. Okay, so I'm going to line them up. That's what happens. My chromosomes line up in the middle. Okay, and then, okay, you get these spindles which start to pull those chromosomes to each side. Okay. Okay. And now this bit here, okay, where you go from the nucleus kind of splitting apart, lining up and splitting those, this is really what they're going to refer to as mitosis. I like to think the whole thing is mitosis, but this is what they're going to refer to as stage 2. Okay, that kind of splitting of the nucleus and the chromosomes moving to each side. Well, then what happens is this cell membrane starts to pinch in like that, and I get my chromosomes on each side. They then split to form, and we get the nucleus reassemble, and we split to form 2 identical cells to what we started off with. Okay, now again, that's really logical. If I was again going back to my photocopying analogy, now I've put them in each piles. I know I've got page 1 on each side. I can then just collect up the piles, and I've now got 2 perfect copies of my exam paper. So, describe what happens. Well, firstly, you can just draw this diagram. Okay, don't hesitate about drawing diagrams. So if I drew this, I'd actually get most of the marks, as long as I annotated it and I labelled it. But I'm going to put it into words, Okay, just to make it a little bit clearer. So, stage 1, okay, that's where here we had the cell grow, we had the DNA replicate and stuff like that. Okay, so the cell grows and replicates the DNA and other organelles, because I'm making a full copy to then split in half. Stage 2, okay, is where we now get the nucleus disappear and the chromosomes start to move to each side. So nucleus disappears, the chromosomes line up in the middle, and one set moves to each side. And then stage 3 is where that cell membrane started to divide the cell. So the cell membrane splits the cell into 2 new identical cells. Done. Okay, last question. So, figure 10 shows how embryonic stem cells are produced in therapeutic cloning for use in patients. Give 2 advantages and 2 disadvantages of therapeutic cloning in medical treatments. Okay, so here I've got a donated egg cell and I've taken out the egg, so I've now just got the egg cell. I take the body cell from the patient and I take the nucleus of it. Okay, I put that nucleus into the egg cell, and then what happens is that egg cell divides into an embryo. Some of those are cultured and some of those are not used. Okay, well, if you notice, all of the genetic information comes from the body cell of the patient. Okay, so that means that any stem cells produced are going to be from that patient, and that means that they're not going to be rejected by the body, or they're unlikely to be. Another thing that's good is the fact that what actually can stem cells do? Well, they could have the potential to cure specific medical conditions, things like paralysis, things like heart disease, things like diabetes. Okay, where your body can't repair the cells that it has. So if you introduce stem cells, you give the body the ability to create those cells which it can't, and therefore cure the disease. So for instance, in paralysis, you could repair a nerve cell, and therefore you've made that connection again, and therefore that person is no longer paralysed. So advantage 1 is stem cells can treat many, can potentially, because a lot of this is kind of still developmental, many diseases like paralysis or diabetes. And the advantage of this system is that you're using the patient's own cells, so you've got unlikely to be rejected, as the person's own DNA is used. Okay. Some other advantages. Okay, if you're using your own body cell from the patient, then you're growing organs essentially from nothing. Potentially, that means that you don't have to worry about organ transplant lists or anything like that. You could use them for not just in treatments, you could use them for research and all sorts of other things. Okay, now disadvantages. Big disadvantage with the egg cell method is you're harvesting an egg cell. That's not particularly pleasant. There's not many egg cell donors out there, and also there's kind of an ethical dilemma about the idea that an egg cell could be fertilised by a sperm cell and create a new life. Okay, so those are the big ones there. Other things, this is quite new and untested. Don't know if it could cause a tumour or something like that. Also, this process is really hard. Okay, the first cloned mammal was Dolly, the sheep, and they did this many times in order to actually get this this nucleus into the egg cell to actually start to divide. So, disadvantages, okay, is that a potential life is destroyed. Debatable, but there you go. Something which people are obviously concerned about. Disadvantage 2 is, I would say, something about the difficulties of egg donation. Okay, because this is a donated egg cell, so egg donation is difficult. Okay, basically you have to give fertility drugs and use a giant needle. Has risks, and there's also a shortage of donors. And that's the end of the paper. Okay, I don't think that was too bad. Hopefully, again, you find this useful. I think the most useful way of doing this is to get the exam paper in front of you and follow along with what I'm doing. As I said, I'm using that first part from the foundation paper, so apologies if the question numbers are a bit jumbled up. It's all the same questions, I promise you. But I hope you found that useful. Again, that was the Combined Science 2018 higher tier, and thanks.